Hello everyone, my name is Evelise Wong, and today I will be presenting on the article Microbiome Metabolite Linkages Drive Greenhouse Gas Dynamics Over a Permafrost Thaw Gradient. So starting off, we'll start off with the bottom line up front. So the main point of the article is aimed at how the interactions between microbiomes and their metabolites affect greenhouse gas emissions in a permafrost gradient. The importance of the research stems from the fact that not much is known about the effects that the microbiome has on greenhouse emissions and what the effect of permafrost thawing at an increasing rate, which is releasing greenhouse gases, can have on the environment and climate overall. So a major concern with this is that methane and carbon dioxide are actually released into the atmosphere from this thawing permafrost and can further accelerate climate change. And so the main thing is trying to figure out how these microbiomes affect this greenhouse gas emission and if there's a possible increase in the rate of greenhouse gases being released. So starting off with a little background, the study and research is done at the Stortalen Mire. So it's a mire in Sweden. It serves as a model permafrost ecosystem in northern Sweden. So in this mire, there are three thaw stage habitats. The dry permafrost underlane of palsas. Palsa is just a peat mound with a permanently frozen core. And then there's the partially thawed and inundated ombrotrophic bogs. Ombrotrophic, it just means that the bog is solely dependent on precipitation such as rain or snow for water and nutrient supply rather than groundwater or streams. And then there's the fully thawed and inundated minerotrophic fens. And so minerotrophic is just, it just receives water and nutrients from mineral-rich groundwater or surface water. And so with the thaw progression that occurs, it actually alters the microbial communities, the vegetation, and soil organic matter composition, which then results in an increased greenhouse gas emission. So we're going to go over a few key terms. There's three main categories: deterministic, stochastic, and then there is the undominated, undominated. And so starting off with the deterministic, there's homogeneous selection and variable selection. And so homogeneous selection from a microbial perspective is just a steady selective pressures originated from persistent environmental condition, which are the main cause of low compositional turnover between a pair of local communities. And then from a metabolome perspective, it's just common selective forces which lead to similar metabolome assemblages. And then as far as the variable selection, from a microbial perspective, it's a divergent environmental factors which cause high compositional turnover between pairs of communities. And then the metabolome perspective is systematic differences among metabolite production and degradation rates, which cause changes in metabolome composition. And so with the homogeneous selection, it's just when there are more ecologically similar communities than are expected by change. And then when it comes to the variable selection, variable is more ecologically dissimilar communities than expected by change. And so then on to the stochastic. So there's homogenizing dispersal and dispersal limitation. And so in the homogenizing dispersal, as far as the microbial perspective, high dispersal rates cause mixing between the communities. And then the metabolome perspective, high rates of metabolite exchange can lead to homogeneous metabolomes. And then for dispersal limitation, from a microbial perspective, low dispersal rates combined with weak or unstructured selective pressures that enable ecological drift, leading to high compositional turnover between a pair of communities. And then as far as the metabolome perspective, low rates of metabolite exchange and unstructured environmental conditions enable the equivalent of ecological drift to cause high compositional turnover between a pair of metabolomes. So with homogenizing dispersal, there's little compositional turnover between a pair of local communities in a microbial perspective. And then as far as the metabolome perspective, it's passive movement by vectors or physical forces. And so now onto the undominated, the microbial perspective. The compositional turnover between a pair of communities is not dominantly driven by selection, dispersal, or ecological drift. And then the metabolite perspective, the compositional turnover is the compositional turnover between a pair of metabolomes is not dominantly driven by selection, movement, or the equivalent of ecological drift. And then another key term is the beta nearest taxon index. It's a standardized measure of the mean phylogenetic distance to the nearest taxon between samples or communities. And this is often used in ecological studies to understand how communities assemble. And this is especially done in soil research. And so the beta nearest taxon index, if it's less than -2, it's considered homogeneous selection. If it's greater than 2, it's considered variable selection. And values between -2 and +2 indicate a randomly dispersed phylogenetic structure driven by stochastic effects. And so just to emphasize, stochastic effects is like a randomized system. It's undetermined. And so now a little overview of the article. So in the article, it examined the agreements between microbial communities and metabolomes in community and feature scale. And so what it means by community is microbial lineages, and then feature scale is the metabolite formulas. And so what they had hypothesized is that the synchrony in ecological assembly processes at the community level signified a stable, coordinated ecosystem response, while feature scale alignment suggests specialized, finely tuned microbial activities and metabolite patterns, which we'll later find out is not the case. So as far as sample collection, so samples of the peat soil were collected at 3 depths, being shallow, middle, and deep depth, from the Pausa, the sphagnum, and the sphagnum-dominated bog, and the Areoforum-dominated fen. And so these cores were collected using a circular push core, and this was done in triplicates. In order to avoid cross-contamination, 1 centimeter of each core's edge was not included in the sampling, and the core was rinsed in distilled water between cores. And so the samples were then transported on dry ice until stored at -20 degrees Celsius until analyzed. And then the ones that were for microbial analysis were placed in cryotubes mixed with a solution called Lifeguard solution by MoBio and stored at -80 degrees Celsius. And so what the Lifeguard solution does is that it's used for the collection and transportation of soils needed for RNA or DNA extraction, in which it protects the viability of the bacteria in the soil while keeping them in stasis. And now on to the methodology of the study. So what they had done was use direct infusion Fourier transform ion cyclotron resonance mass spectrometry. And so what this does is that it leverages the high resolving power and ultra-high mass accuracy and sensitivity of FTICR MS with the rapid data acquisition from direct infusion into the mass spectrometer ion source. And so this was used to gain a comprehensive overview of the soil metabolome across the thawing permafrost gradient. And so since the soil organic matter or material is comprised of complex and dynamic mixture of metabolites, they decided to use this method because it detects a wide range of individual molecules for characterization of molecular composition and transformation. Another benefit of them using this was the wider detection of low abundance metabolites without having to focus extensively on structural annotation. And then in addition to this, they used the DNA extraction and sequencing of the samples using the Power Max total nucleic acid extraction kit, once again by MoBio. And then this was sequenced with HiSeq and NextSeq platforms, which is done by Illumina. And so then they also did the Agilent 2100 bioanalyzer and the Agilent 220 tape station, which was used to test the quality of the RNA and the RNA libraries. And then to measure RNA quantity, they used the qubit. And then back to the Illumina sequencing, samples were sequenced on 1/8 of the next sec with shallow runs conducted on the 1/11th of a high sec in MiSec. And so the benefits of using Illumina sequencing is that it has the ability to generate large amounts of data with high sequencing output. The only issue with this is that the short read lengths can make it difficult to assemble complete genomes of microbes that may have not been found previously in the permafrost or are not well cultivated. And so once again, the RNA extraction and sequencing was done by Illumina in order to generate data to provide insight into the metabolites and microbiomes of the peat material and how it affects the environment. So starting with the results, with Figure 1, the ecological assembly processes driving active layer permafrost microbial communities and metabolites. So what they had found out was that during the investigation into the shaping of the microbiome and metabolomes, it was shown that the microbial assembly shifted along the permafrost thaw gradient from environmental filtering and biotic interactions towards a more increased stochasticity, resulting in low compositional turnover with habitats. And this can be seen in the violin plots in A and B. And so on the A side, you have the microbiome. On the B side, you have the metabolome. And so what this plot does is it shows the average within the habitat of the beta NTI. And once again, this is an index used to compare the ecological processes driving microbial and metabolite assemblages. So in this, the microbial assembly was driven by homogeneous selection in the palsa and homogeneous dispersal in the bog and fen. And this can be seen in part E of this figure. In the palsa, limited spatial turnover was most likely due to the consistent environmental filters, such as the dry ombrotrophic conditions, and then also there is very strong seasonal freezing thaw cycles that occur here. And so then another thing is that increased water availability actually promotes dispersal in the bog and fen. So due to these various ecological factors that shape bog communities, such as the sphagnum presence, the low pH, the nutrient levels, and water table fluctuations, this diverse soil organic matter composition provided various avenues for microbial communities to thrive. And in contrast to the microbial assembly, metabolome compositions were primarily driven by variable selection and homogenizing dispersal. So it was shown that the bog has the highest variable selection, followed then by the palsa and then by the fen. But the homogenizing dispersal had the most impact on the fen, and then after that was the palsa and the bog. So they were almost kind of flipped in a way. So despite what was hypothesized, the sphagnum-derived metabolites did not create a uniform metabolome. It was actually more by variable selection, which suggested that that microorganisms can degrade polyphenolic compounds. Another thing is that, as seen in the in C and D, which are density plots, this showed changes in the beta NTI of each habitat, and this was plotted with the depth in the month. And so then in E, back to the bar plots, it actually shows that there's an influence of different ecological processes within each habitat according to the microbial community and bulk metabolites. The heat map shows that the Mantel correlations between peat samples of the beta NTI calculated within bulk metabolites and the pairwise differences of each environmental variable between samples, that these values range from 1 to -1, which both shows a positive and negative correlation. In addition to this, the carbon to nitrogen ratios in precipitation actually significantly contributed to shaping the pulses in the fen metabolomes, which ends up showing that the influence of oxygen availability, microbial activity, and peat stratification on the metabolome assembly. So it shows how oxygen can affect this. So even though the bog experienced a higher influence of variable selection, there were actually no significant correlations with environmental factors, meaning that there's still like unknown variables at play. So that's something that still needs to be looked into because there's something affecting that they just have not found out yet. And then so another thing was that the strongest connection with environmental variables was observed in the fen impulses. And so this suggested that the bog itself is potentially organized at the feature scale, once again, and then driven by connections of individual features. So overall, this highlights the importance of the feature scale microorganism metabolite connections in the bog, and then also the need for different approaches to understand these processes that are shaping the permafrost ecosystems. So Figure 2, metabolite beta NTI feature-derived clusters correlate with specific microbial taxa and environmental factors. So with this, it showed strong positive and negative correlations between genomes abundance and the metabolite clusters in each habitat due to the peat spatial complexity, which created microbial niches. And so Bog cluster 1 is a prime example of this, in which the sulfur-containing metabolite clusters with CHOS elemental compositions were actually linked to the metabolome. What this did was that it hinted that the sulfur-containing metabolites actually play a key role in soil organic matter cycling within the bogs. And then when comparing the metabolites across plant species and peat soil from the 3 different habitats, it was seen that the sphagnum had a lower NOSC than the other plants. So another thing is that sulfur-containing compounds were highly consumed in the bogs, and compounds observed in the plant extracts or the peat were abundant in the bog and fen. So overall, findings had shown that the bog microbial communities actually co-evolved to use these metabolites in an efficient manner. So on to Figure 3, correlation of genomes that significantly correlated with the beta NTI feature metabolite clusters with carbon dioxide and methane in the bog pore water. And so with this, there were significant correlations between genome abundances and the greenhouse gas levels that were observed only in the bog. So this was done with 45 metagenome assembled genomes, also known as MAGs, which correlated with carbon dioxide, and 29 of these MAGs had correlated with methane. And these MAGs were compiled from 2011 to 2017. And so in this figure, A and B actually focuses on the 5 of the genomes that were found to significantly contribute to community assembly. So the lack of significant correlations in other habitats probably suggests that the relationship between ecological processes and biogeochemical functions may occur at different scales along the thaw gradient. Specifically in the bog, where there are strong ecological filters such as the low pH and the presence of recalcitrant compounds seem to contribute to more greenhouse gas production. So this is different in the pulses and in the dens. So as far as the pulses and dens, the fens, this function may depend on the action of many members of the microbial community, making their contributions less important at the feature level compared to the bog. And so then in part C of this figure, this actually shows the functional potential of these taxa. And so this is a heat map, and this heat map is divided into functional categories on the right with different functions with each category. And then the color rectangles are the presence of a function that is observed. And so of the 5 bacterial genomes that correlated with greenhouse gas levels in the bog pore water, 2 of those genomes, which was the Pteroacidophilus and the Fen 455, which one is a Acetobacteria and one is Actinobacteria. These both had positive correlation with dissolved carbon dioxide and methane concentrations. And then Holophagia, which is Acetobacteria, correlated solely with carbon dioxide. And then RAAP2, which is Actinobacteria, correlated negatively with carbon dioxide and methane. And so another thing to note is that Actinobacteria actually, their genomes express genes for producing and utilization of methane substrates. And so back to the Pteroacidophilus, these genomes expressed a wide set of enzymes for the degradation and utilization of polysaccharides, which highlighted their role in breaking down biopolymers and contributing to core biogeochemical processes. And so another thing about the Acetobacteria is that they've been described as the primary degrader of sphagnum polysaccharides in the peats and bogs as well. So figure 4, microbial metabolite co-occurrence networks within each habitat across the thaw gradient and their functional potential. So with this, the bog, which is B there, the bog exhibited the most microorganism metabolite links, followed by the fen and the pulsa. So this actually corroborates with the analysis that the bog has a strong feature scale organization once again. And so so those top images there, A, B, and C, they actually show networks within the pulsa, the bog, and the fen in that order. So A is the pulsa, B is the bog, and C is the fen, in which metabolites are represented by circles and microbial phyla are represented by squares. And so the larger circles or squares actually represent microbial and metabolite node hubs and connectors. These nodes in all 3 habitats were all lignin-like compounds that serve most likely as like a primary nutrient source for microorganisms. And so then on the bottom there, D, E, and F are the alluvial plots. And so these plots are led to believe that microbial community contributions to metabolic and biogeochemical processes are actually notably in the pulsa and the bog. So these plots also represent the contribution of different mags to individual metabolic and biogeochemical processes within carbon, nitrogen, sulfur, and other cycles in the pulsa, the bog, and the fen. So the genomes within each habitat are actually represented at the phylum level. And then there's 3 columns from left to right, which represent the taxonomic groups scaled by the numbers of genomes and the contribution to each metabolic functions by the microbial groups. And then the different colors just represent different phyla. Okay, so contrary to the initial hypothesis of congruence between microbial communities and metabolites, it actually showed that there was a divergent assembly pattern across the thaw gradient, which was observed. So, they were not leading to a more stochastic situation, but that turned out to be what the case was. And so then another important finding was that organosulfur compounds potentially originating from sphagnum mosses were identified as important factors that influence both the metabolome composition and microbial community assembly, which impacts the greenhouse gas composition of the pore water. And then it also highlighted how sulfur and carbon cycling dynamics in peatlands when sulfur cycling exerts an important control on organic C, which is carbon degradation and greenhouse gas emissions. And then overall, what was revealed was that there was a decoupling between drivers of the microbiome and the metabolome. So instead of it being congruent like they had originally thought, it was the opposite. And then it also had showed that the microbial assembly shifted along the permafrost thaw gradient from a more environmental filtering and biotic interaction towards the increased stochasticity, which once again is just it's the system. It had shifted to a more unpredictable system. Uh, Biggest thing is that there's still much unknown about microorganism metabolite interactions, the permafrost gradient. So more work needs to be done on that, as far as even on a molecular scale, or just future studies as to how this could affect the greenhouse gas emissions, especially since we see that there are certain bacteria that can utilize methane. And so I think it could be potentially important in the future to figure out if we can increase those levels of that type of bacteria, if we can reduce the rate of greenhouse gas emissions. So here are a couple questions. Question number one would be, could the Eriforum found in the fens play a similar role to the sphagnum moss found in the bog regarding greenhouse gas emissions? 2, would the release of these greenhouse gases from thawing permafrost lead to a positive or negative feedback loop in the overall climate? 3, if the permafrost completely thaws, what kind of changes in the microbiome and environment could be expected? 4, what techniques other than DI, FTICR, MS could be used for metabolite identification in the thawing permafrost? 5, as the permafrost continues to thaw, which group of bacteria is most likely going to be dominant between the acetobacteria and actinobacteria? And then here are my references, and thank you everyone.